Hello my dear friends, I'm Sol from Biological Sciences with Sol channel. Today I'm with new video of the control of gene expression and mutation, which is part of genetics and evolution. Today I'm really happy because my YouTube channel is accepted as YouTube partner program and it's going to be monetized. I would like to thank you all, and as usual, don't forget to like and share this video and subscribe this channel, and let us proceed to the main part of this video. Control of gene expression. So first let us know gene itself. As you know, DNA is large or huge molecule which contains different sections. So these different sections of DNA are known as genes. So each of these genes determine particular characteristics of an organism. They determine the characteristics of an organism through expression to protein, because DNA is transcribed into messenger RNA. That means genes in a DNA are expressed or transcribed into messenger RNA, then messenger RNA is translated into protein. Therefore, genes determine the characteristics of an organism through expression to protein. So, synthesis of protein under the influence of gene is known as gene expression. Genes in organisms expressed in different manners. All the genes of a cell are not expressed at all the time, even if same gene is found in different cells, but they expressed in different manners. For instance, insulin gene can be found in all the tissues. However, it is expressed only in the beta or insulase of Langerhans cells of pancreas, but not in other tissues. In other words, insulin gene is under or in the state or condition of repression in other cells, but it is expressed in the insulase of Langerhans cells of pancreas. Still, there are some genes that are expressed in all cells. For example, genes that are expressed into enzymes of glycolysis are such type of genes. As you know, glycolysis is a pathway through which glucose is oxidized. For this purpose, a number of enzymes are required. So these enzymes are formed from genes that are expressed in their respective enzymes in all the cells, because this glycolysis occurs in all the cells of the body. So such type of genes are known as constitutive genes, or they are also known as housekeeping genes. Still, in other hands, the same gene may be alternatively open and shut as per the need for the metabolism. Such regulation is done by induction and repression mechanisms. Induction turns on and repression turns off the switch of gene expression. Induction is the phenomenon of increased synthesis of protein or enzyme in the response of certain signals. Such enzymes are said to be inducible enzymes, and the signals are also known as inducers. The next point is about mutations. This mutation is the alternation of the nucleotide sequence of genome of an organism, a virus, or extra chromosomal DNA. If there is a change or alternation of nucleotide sequence of the genome of an organism, then the resulting protein will be different type, then organism will be affected. So there are some properties of mutation. For example, mutation occurs spontaneously or suddenly during DNA replication or DNA duplication. Second, mutations are rare events. For instance,
Each cell contains 6 billion base pairs. Among these, only on average 120 mutations will be occurred. That means on a ratio 1 to 15 million. Most mutations are detected and repaired. Even 95% of our DNA is non-coding DNA, or the genes on DNA. Almost 95% of the genes in DNA are non-coding genes, so mutations are likely to affect the coding genes. There are factors, or there are conditions that have increased rate of mutation. For example, carcinogenic chemicals such as chemicals in tobacco smoke increase rate of mutations. Even high energy radiations such as ultraviolet radiation and X-ray also increase rate of mutation. Type of mutation. There are two main types of mutation. One is point mutation, and the other one is chromosomal mutation. In point mutations, changes that involves a single base or a single nucleotide. If, of course, this point mutation also include substitution mutation, addition, and deletion mutations. So we'll see in the next slide addition and deletion mutation. So in substitution mutation, one base is replaced by another base. If one base is substituted or replaced by another base, so the mutation type is known as substitution mutation. For example, here in base sequence of DNA, adenine, thymine, guanine makes the first code. Guanine, thymine makes the second code. Thymine, cytosine, adenine the third code, and guanine, adenine, guanine make the fourth code. These base sequences in DNA are transcribed into corresponding base sequences in messenger RNA. Therefore, adenine, thymine, guanine is transcribed into adenine, uracil, guanine. Thymine is replaced by uracil because it is base sequences in messenger RNA. Then guanine, thymine codon transcribed into guanine, uracil. Thymine, cytosine, adenine transcribed into uracil, cytosine, adenine, and guanine, adenine, guanine, same guanine, adenine, guanine. Then these base sequences in messenger RNA are translated into amino acid sequences. So adenine, uracil, guanine codes for methionine. Guanine, uracil codes for glycine. Uracil, cytosine, adenine codes for serine, and the guanine, adenine, guanine codes for glutamate or glutamine. This is the first polypeptide chain, or it is corresponding amino acid sequence. If guanine in the second codon of messenger RNA is substituted by cytosine, then it will be adenine, thymine, adenine, similar, guanine, cytosine, thymine, because this cytosine replaces the guanine in the second codon, then thymine, cytosine, adenine, guanine, adenine, guanine. Then this adenine, thymine, guanine is transcribed into adenine, uracil, guanine. Then this guanine, cytosine, thymine is transcribed into guanine, cytosine, uracil. Thymine, cytosine, adenine to uracil, cytosine, adenine. And finally, guanine, adenine, guanine to the same guanine, adenine, guanine. And this messenger RNA codons are now translated into base sequences. So adenine, uracil, guanine to methionine, guanine, cytosine, uracil to alanine. Here, in the second polypeptide chain, the second amino acid is alanine. whereas in the first polypeptide chain, the second amino acid is glycine. So glycine is substituted by alanine because of substitution of glycine by cytosine. So immediately the formed polypeptide chain is changed due to replacement of glycine by alanine due to a replacement of guanine by cytosine. So this type of mutation is known as substitution mutation. Addition mutation, which is a type of point mutation, if an extra base is added. Here, for example, the first base sequence or the first code is adenine, thymine, guanine, then guanine, thymine, cytosine, adenine, and the guanine, guanine. If adenine is added, one base is added in between these two guanine, then immediately instead of guanine, thymine, it is guanine, adenine, guanine. Then this thymine will be involved, or it is included in the third codon. So thymine, cytosine. Now this adenine, which was in the third codon, now taken into adenine, guanine, adenine. Then this guanine will be left alone. So this type of mutation makes a frame shift. Plus, this deletion mutation is also point mutation type if a base is missed out during DNA replication. If guanine is deleted from the second codon, here if guanine is missed or lost, then the result will be adenine, thymine, guanine, then guanine, instead of guanine there is thymine, then thymine. This is the third, thymine is the third codon or code will be shifted to the second. So there is still frame shift frame shift occur. Therefore, addition mutation and deletion mutations are also known as frame shift mutation. Chromosomal mutations are another type of mutations, a situation where part or segment of chromosome is disturbed or a chromosome is missed or added. So at this time, chromosomal mutation occurs. Some chromosomal mutation include euploidy or polyploidy. This time, gaining of one or more complete set of chromosomes. For example, our body cells have diploid types of chromosomes because they have 46 chromosomes. That means 2 times 23. This N means 23. But in euploidy or polyploidy condition, organisms may have 3 N types. That means triploidy, or they may have tetraploidy, 4 N type. Because one complete set of chromosomes, that means 23 chromosomes are added in case of triploidy. In tetraploidy type, 46 chromosomes are added. So this type of chromosomal mutation are known as euploidy or polyploidy. Aneuploidy is another chromosomal mutation. If one or more single chromosome is added or lost, this aneuploidy is formed. For example, trisomy is a good example of aneuploidy. If one single chromosome is added and 47 instead of 46 chromosomes are found in a single body cell. A good example is Down syndrome. This Down syndrome is characterized by low muscle tone, short stature, a flattened nasal bridge, and a protruding tongue. The other type of aneuploidy is monosomic. So here in monosomic, one chromosome is lost. So instead of being 46, there are 45 chromosomes are found in a single cell, body cell of human being. For example, Turner syndrome is a good example. This Turner syndrome is mainly occurred in female when they lose one sex chromosome, and it is characterized by short height, failure of the ovary development, and heart failure or heart defects. The other type of aneuploidy is double trisomy. If there are two chromosomes are added, so instead of being 46, if there are 48 chromosomes are found. And the other type is nullisomic. If two chromosomes, especially these two chromosomes are homologous chromosomes, are lost. So instead of being 46, 44 chromosomes are found in a body cell. So a good example of this nullisomic are Goody syndrome. This Goody syndrome is manifested as mental retardation, which is caused by deletion of chromosome number 5 of human. This chromosome number 5 means the two chromosomes, the homologous chromosomes. Consequences of gene mutations. By the way, we have two types of cells, body cells and sex cells. So mutations that occur on the sex cells directly inherited to the next generation, so it affects the offspring. But mutations that occur on the body cells cannot be inherited, but results in one of the following. It may be harmless, it may damage or kill the cell, it may make the cell cancerous, which might kill the person. There are two types of genes that are important in regulating cell division and preventing formation of a tumor or cancer. The first gene is proto-oncogene. When these genes mutate, they become active oncogene. So this active oncogene stimulates cell division in uncontrolled manner. Then tumor suppressor gene recognize this uncontrolled cell division and acts to suppress cell division. If tumor suppressor gene mutates, it becomes inactive, then tumor will be formed as uncontrolled cell division continues.
Thank you once again for watching, and I will come back soon with another new video, and see you.